So today we are going to be talking about DNA, also known as deoxyribose nucleic acid or deoxyribonucleic acid, which is just taking these letters away, the S and the E. Now the model for DNA was proposed by James Watson and Francis Crick in 1953 and was accepted by scientists when it was proposed. Now the structure of DNA, the one that they proposed, is the 5-carbon sugar, found right here, a nitrogenous base, and a phosphate group. Now, the 5-carbon sugar, what you need to know are that there are carbons where the primes are, the numbers like 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. And they are important to the structure of DNA. And the nitrogenous base consists of A, T, G, and C. Also U, but that is only important for RNA. Adenine and guanine are purines, meaning that they are double ringed. So they have 2 rings. Thymine and cytosine are pyrimidines, which means that they only have 1 ring. So instead of this extra ring, they would only have this one. They only contain 1 ring. Another important thing about the nitrogenous bases are that adenine always connects with thymine, and guanine always connects with cytosine. They are pairs, so G and C are a pair, as well as A and T. Now, this is how DNA connects with more DNA. Here we can see that we have a nitrogenous base, a 5-carbon sugar, and a phosphate group, but there are more. Now, the way they connect are through hydrogen bonds, and a hydrogen bond is just a connection made with water. So, as we see here, we can see that there is a connection in 3 prime, and there will always be a connection at 3 prime when you connect DNA. So, if we are trying to create water, we are going to lose water in order to connect this and this. And in order to make water, we need 2 hydrogen and 1 oxygen. So, what we will do is lose 2 hydrogen and 1 oxygen, leaving 1 oxygen in order to form that hydrogen bond. And now the same applies to this phosphate and this 5-carbon sugar, and it is just a repeating chain. Another important thing you need to know is that the order of this strand is 5 prime to 3 prime, meaning that whichever prime is closest to the top is what the top will be labeled, and whatever is closest to the bottom is what will be labeled at the bottom. So here, if we number all of the carbons again, which are just the corners that have not been labeled, it would be 1 prime, 2 prime, 3 prime, 4 prime, and then 5 prime, because there is a carbon right here. And this end is closest to 3 prime. This end is closest to 5 prime. If we number them again, we can see 1 prime, 2 prime, 3 prime, which we already numbered, and 4 prime. And that is how this strand is constructed. Now, we know that DNA has 2 strands, not just 1. So how is the second strand compared to the first strand? Now, if we take a look at the first strand, we know that it is 5 prime and 3 prime. And we also know that DNA is anti-parallel, meaning that it is just the inverse of what this is. So if we copy this and paste it, all we have to do is just flip it. So then the nucleic acids can form bonds as well. Now, here's where we have to get a little specific. So let's label these nucleic acids. The first one we can label as A, adenine. The second one we will label as thymine. And the third one we will label as guanine. Now we know that adenine and guanine are purines, but thymine is a pyrimidine. So let's get rid of the second ring. Now, let's pretend we don't know what the labels of these are. So, with what we know is that A always pairs with T, and G always pairs with C. So, we can just label the second strand. And the second strand we know will be T. It would be single ring, so let's just go ahead and get rid of the second ring. Thymine always pairs with A. So we can just go ahead and write that here because we know that A is a purine, so it is double ringed. And now G pairs with cytosine. And cytosine is a pyrimidine. So that means we have to get rid of the second ring. Now, not all of the nucleic acids have the same chemical formula. But for the basis of this video, we will be using adenine, which is the formula you see right here. But in general, pyrimidines only contain one ring, which you can see here, here, and here. Purines, two rings, which is adenine and guanine. And now, if you can take a look, since we took the inverse, which is just the opposite of the first strand, we can look at where the primes are. So now, since we flipped it around, the top should be 3' and the bottom should be 5', because 5' is right here, 4' is right here, 3', 2', and 1'. Now if we label it over here as well, 5', 4', 3', 2', and 1', the closest to the top is 3'. The closest to the bottom is 5'. Now, if we take a look at these nucleotides, which are just guanine, cytosine, adenine, thymine, and adenine and thymine, how are they connected? They are connected through hydrogen bonds. The important thing you need to know about it is that the hydrogen bonds between these differentiate. Now A plus T will have two hydrogen bonds, meaning that they connect twice through hydrogen bonds. Now this first dashed line is one hydrogen bond. The second one is another hydrogen bond, and that applies to the T plus A. As for G and C, it actually contains 3 hydrogen bonds. One, 2, and 3. And that is all for the overview of DNA.